I never meant to call you when you sorrow. I never meant to call you when you pain. I'm the one who wants to see you laughing. I only want to see you laughing in the purple rain. Purple rain, purple rain. Purple rain, purple rain. Purple rain, purple rain. Only want to see you bathing in the purple rain. I never wanted to be your
Okay, thank you to Prince for that lovely introduction. Today we are talking about color vision. Yes, we are. All about color vision today. And I will come back to why Purple Rain was playing earlier. That has some significance today as we talk about color vision. To get started, I'd like to go back to a little bit to the very first time we started talking about vision, to talk about the dimensions of color, right? When we talked about the physical stimulus for vision, we mentioned this, but color or hue is going to be determined by the wavelength of the light. So short wavelengths are blue, medium wavelengths are in the green range, and then long wavelengths are in the red. And of course, we have lots of hues in between. The hue, the color of the light, can be attributed both to direct light and to reflected light. And we'll talk about the differences between those. White light contains all the wavelengths, right? So equal stimulation of short, medium, and long wavelength cones, the three photopigments we have, equal stimulation produces the sensation of white. So the first question that I have for you is, why is the sky blue and the sun yellow? Can you figure that out? Any clues? This is where if we were in class, I would be happy to take some answers on this. But it turns out the reason that the sky is blue and that the sun is yellow is because the Earth has an atmosphere. And in that atmosphere are gas molecules and other particles. And those molecules scatter the short wavelengths of light so that they're reflected and we see the short wavelengths of light more than the medium or long wavelengths. So blue gets scattered and we see the sky as blue. And then if we look at the sun, what wavelengths are left, right? So the wavelengths that are left are medium and long. And if you have equal stimulation of medium and long wavelengths, you get yellow. That's in between green and red. So that's why the sky is blue and the sun is yellow. So when your 3-year-old niece or nephew asks you that question, you now have a legitimate answer that you can give them, right? So as we see with this prism, if we take white light or all the wavelengths of light and we scatter them, we can separate them out into their various hues. All right, next question. Why are clouds white or sometimes dark gray? What is in a cloud? That's right, moisture, right? Moisture is in a cloud, water. And it turns out when we have finely dispersed water droplets in a cloud, they are scattering all the wavelengths of light, all the wavelengths of light equally. So therefore we get white. When do we see really dark gray or even blackish clouds? That's right before it's supposed to rain, right? Rain clouds are very dark. That's because they're so densely packed with water that the water's going to precipitate out of the clouds and fall to the earth as rain, right? But they're so densely packed that instead of reflecting all the wavelengths, they start to absorb the wavelengths. So the absence of light is going to be black, right? Or gray, or a darker shade of in between white and black. All right, So clouds and fog is moisture particles. When light hits them, it's going to equally scatter the light and appear white, right? Okay. Is the sky always blue? No, it's not. At night, it's black because there is no light, right? But in between the daytime and nighttime, sometimes we have really pretty colors in the sky, like pinks and reds and oranges. Why do you think that is happening? It is because when that only happens at sunset and sunrise, right? And so if we look at the sun directly above the United States, right, the amount of atmosphere that the beams are traveling through is very small compared to the amount of atmosphere that the sunlight is going to travel through when the sun is rising or setting. So if we move the sun over to Europe, so this would be like sunrise for us, middle of the day in Europe, sunrise for us, you can see the wavelengths are traveling through much more atmosphere. Here's another depiction of that, right? So when the wavelengths are traveling through much more atmosphere, we start to scatter not just the short wavelengths, but now we start to scatter the medium and the long wavelengths. And so we get yellows and oranges and reds if it's mostly long wavelengths. And sometimes we get purple. We'll talk about that in a second. What color is the sky in outer space? So if you were on the space station, or if you've seen pictures of people doing a spacewalk, right? What color is the sky in outer space and why? Or what color is the sky on the moon? We could say that as well. It's black, right? So why is it black? Because there's no atmosphere, there are no molecules to scatter light, so the wavelengths are just passing straight through the void and not being scattered. The moon doesn't have an atmosphere, therefore the sky always looks black on the moon. Okay, what color is the sky on Mars? Have you ever seen pictures from the Mars rover of the Mars landscape? I'll give you a hint. It's a nickname for Mars, the blank planet. Yes, it's red. Now, why is it red? It's red because Mars has different gases and different molecules in its atmosphere that scatter long wavelengths of light and let the medium and short wavelengths of light through and selectively scatter the long wavelengths of light. So here's the even trickier question, right? If you're on Mars and you look up at the sun, what color is the sun going to be? It's actually going to be a combination of short and medium wavelengths of light because that's what's shining through. The red is being scattered in the sky. So it's going to appear like a teal color, a mixture of blue and green, which is really freaky, kind of weird to think about. Okay, so if the Earth's atmosphere generally scatters short wavelengths of light, right? What about if we see big objects that are far away, like skyscrapers in the distance or mountains in the distance? They take on a bluish tint, right? So we call them the Blue Ridge Mountains, or in the song Purple Majesty, right? And the mountains really aren't blue. Right? If you get up close to them, they're green, usually with the foliage. Or even sometimes, if it's fall, they're red and orange and all sorts of things. But from far away, they always have this bluish tint because there's so much atmosphere in between us and the faraway mountains that the light gets scattered, the short wavelengths of light are scattered, and we see that bluish tint come through. All right, let's talk about ice for a minute. We can talk about light reflection and absorption. So common ice, typical ice that you get in your out of your ice machine, right, is usually white in color, and that's because it's reflecting all wavelengths equally, right, kind of like clouds. However, sometimes we can make ice that has no crystals and no air bubbles and nothing in it to reflect light, and then the ice would appear clear, like the kind of ice you would see in an ice sculpture, for instance, right? Glacial ice is more compressed, and so sometimes that compression causes the ice to reflect blue more, short wavelengths more than the other wavelengths. And so it takes on this bluish tint, particularly deep in the glacial ice where it's really compacted. All right. Let's describe some of the properties of color. So you can open up a box here. Let's, like this yellow box, we'll do this real quick. Okay, so I'm going to take this yellow box and I'm going to open up the fill and say more fill colors. And I'm going to go to this custom box. And so here you could see as I change the red, so here we have equal amounts of red and green. That makes yellow, right? And in fact, what if I make this 150 and I make this 150, right? We get the same yellow, but now it's not as bright, right? So I'm gonna put this back to the brightest it will get, right? And as I take away the red, right, we're gonna see that top square right here become more green. I'm reducing the red, we're shifting from the yellow towards the medium, and so now we are all just green, right? If we want to know what the sun would look like on Mars, we can add in an equal amount of blue and see what color we get. So we get sort of this aqua color. Wouldn't that be weird to see the sun as that color? Okay. If we go back and forth across the top here, right, we get a whole bunch of different shades of colors. We can detect about 200 different hues of those colors. But if we now add in this vertical dimension, which is called saturation, right, for any one hue, we can detect a whole bunch of other hues. For any pure wavelength, we can detect a bunch of other variations of that. And so if we add that in, we get about 1,200 different combinations that we can detect. All right, so color dimensions. Here we see pure yellow light, monochromatic light in the upper left corner, and I'm varying the saturation down towards the bottom right corner, where at the bottom right corner we have desaturation. We have desaturated, we've turned the saturation all the way down, and we lose the ability to detect that color. It becomes washed out, right? And as we get decreases in saturation, we get sort of that grayish appearance. We can also vary the intensity of the light, and that can affect our hue perception as well. All right, so let's talk about some different colors. Here you see a color wheel. And one interesting and important thing to note is that colors that are opposite each other, like green and red or yellow and blue, are called diametric colors. And these diametric colors actually cancel each other out because of opponent processing that we'll talk about in a few minutes. But they cancel each other out. And so when you mix equal parts of yellow and blue or green and red, you get this desaturation and they cancel each other out. Non-complementary colors are going to produce intermediate perceptions. So if we look here at yellow and we look here at red, if we had equal amounts of yellow and red, we would get this intermediate orange color, right? All right, this is another depiction of the color wheel, and we see now the wavelengths of light, starting from short wavelengths of light, blues in the 400s, to greens in the 500s, to reds in the 600s and 700s. Okay? And so again, if we have diametrically opposite colors, they cancel each other out, right? And this is called hue cancellation. So in this block, you can see I've made the top of the block blue, and I told the bottom of the block to be yellow, and then it's going to automatically fade from blue to yellow. And so you get the hue cancellation in this area, right, where it appears gray or white. And then as you move away from that towards more blue or more yellow, we start to see the hue come back in. All right, so why was I playing Purple Rain, other than it's an awesome song by Prince? It's because purple doesn't exist. Do you see the gap here? Right, we go from 400 all the way around to 700, and there's no purple. Purple exists in this gap. It's called extra spectral. It's outside the spectrum of our color. There is no wavelength of light, single wavelength of light, that can generate the color purple. It's made up in our head. It is a color, it's a perception that we create in our brain. And we create it when we mix equal amounts of blue and red. So when we have an equal amount of blue and red, short and long wavelengths of light, when the short cones and the long cones are both active, our brain perceives that neural signal as purple. And people have known about this for a long, long time, that purple was a special color, that it was different than the other colors. So what, if you think about in history, the color purple, what is it associated with? Yes, royalty, right? It's the royal color. It was reserved for the highest authorities. In the church, bishops wore purple. Purple was a color reserved for the highest ranking members of the church, right? And so maybe you can think of some other examples of why purple is a very special color. But now you know that purple doesn't exist, right? All right, so you're going to learn something new in here every day. Color matching. So as I mentioned, if we have blue and red and we mix them, right, we can't generate a single wavelength of light to match that. But for all the other colors, we can, because there are metamers, what we call metamers, that are the complementary colors that are mixing. So here, if we have a test field that's yellow, right, how could we recreate the yellow test field if we weren't going to use yellow light? Could we use two other sources of light to create yellow, right? And we already talked about this, that if we have equal stimulation of medium and long wavelength cones, that generates the perception of yellow. So if we had equal amounts of green and red light, that would actually generate a yellow test field. So it would be the different stimuli, right? So we have 580 nanometer wavelengths of light, and then we have a combination of 510 and 600, but both of those are going to appear the same. We would not be able to discriminate the difference between a single wavelength of light and two wavelengths of light. And that's because they're stimulating the same pattern in our cones. Here we see that effect. We have a red projector and a green projector, and where those projectors, the light from those projectors overlaps, is yellow. And there is no source of yellow, just red, green, and blue right here, right? So that is called a metamer. It's when pairs of colors mimic true wavelengths or a single wavelength of light, okay? So the metamers are red and green, and the metameric match they create is yellow, right? If we unmask the blue light, right, we can see that if we have green overlapping with blue, we get that teal color. If we have all three wavelengths of light, we get white, equal stimulation, right? And down here is our friend purple, that is equal amounts of red and blue light generates purple, right? Now, it's not a mistake that the three primary colors, blue, green, and red, are the three colors, three wavelengths that our short, medium, and long wavelength cones respond greatest to. Remember those tuning curves, the three tuning curves for each of the photopigments of the cones? So any combination of stimulating short, medium, and long wavelength cones can create a perception of any hue, right? The exception of that is that you can't create red by mixing any other color, and you can't create green by mixing any other color, and you can't create blue by mixing any color. Okay. So this is what we call additive color mixtures. We'll talk about subtractive color mixtures in a few minutes, but right now we're talking about additive color mixtures, and this involves light sources, right? Mixing light sources, and those light sources typically are reflected off of a white surface. If the surface is colored, we'll talk about that at the very end of the lecture about color constancy, then that produces a different challenge. This additive effect, as I mentioned, is due to neural integration of the short, medium, and long wavelength outputs, right? So if we have blue and yellow, right, yellow has equal amounts of red and green, right? So we essentially have equal amounts of blue, red, and green. So mixing blue and yellow together is going to generate a white light. It is as if, even though we only have two wavelengths of light, 450 and 600, we're effectively equally stimulating with those two wavelengths of light all three cones. So if all three cones are equally stimulated, our perception is white. All right, subtractive color mixtures. This is what paints and dyes do. Okay, it's different than lights. So with paints and dyes, they are selectively absorbing wavelengths and selectively reflecting other wavelengths of light. So for instance, with blue, we're selectively absorbing yellow, orange, and red and reflecting the shorter wavelengths of light with that sort of teal color, and equal amounts of green and blue. And with yellow, we're selectively absorbing blues and equally reflecting greens, yellows, and reds. So if we were to mix those paints, we're going to add in the subtractive selective absorptions. So by overlapping those two paints, we're going to absorb, the yellow paint's going to absorb the blue, and the blue paint is going to absorb the yellow and red, and what's going to be left is green. The only color left is green to be reflected out. And again, this is all wavelengths of light. We can also talk about that in terms of filters. It's like using a filter, right? So if you have a yellow filter that's going to filter out the short wavelengths of light, and you have a blue filter that's going to filter out the medium and long wavelengths of light, and you combine those two, the only thing left is green, right? This is the same thing that paints and dyes do. They're assuming that all wavelengths of light are going to strike that substance, so we're going to absorb the ones we don't want you to see and reflect the color that we want that object to be. So, additive versus subtractive. With additive, we are neurally integrating combinations of wavelengths on the on the cones, on the three cones. With subtractive, we are selectively absorbing, the surface is selectively absorbing and reflecting only the wavelengths of light that we want to see. Now, the type of light source matters. As I mentioned, we're assuming that the light contains all the wavelengths of light. And so here we see natural light, sunlight, where it's pretty equal representation across the wavelengths. And then here we see an incandescent light bulb, like the old light bulbs with the filament inside. And so if you're in a room that's lit by an incandescent light bulb, it has a warmer, like orangey appearance, because that light bulb produces more medium and long wavelengths of light, and doesn't produce very much short wavelengths of light. So it doesn't have that real stark white, because our short cones are not being stimulated as much as the medium and long. Right? You can actually go to this website and look at different types of light bulbs and what types of spectrums they put out. Okay, so maybe you've noticed that fluorescent light is very different than incandescent light or LED light. All these different lights we have now, halogen lights, they all have a different sort of hue or spectrum of wavelengths that they're projecting, and that causes the objects in the room to appear slightly different colors. So here we see natural light compared to a fluorescent light. This one happens to be called chroma 75. And fluorescent lights have that sort of stark whitish appearance, almost sort of white blue appearance. And that's because they have a very high wavelength of this sort of teal color that spikes due to the gases that are used in that chroma 75 fluorescent light. You can see the orders of magnitude. I mean, here we're looking at about 150 across here, 125 across here for most of the other wavelengths. And then this spikes up to 650, right? So that's why fluorescent lights appear to have sort of that artificial bluish, bright, stark white appearance. All right, why does this matter? Well, it matters because depending on what type of light you're using, it changes the wavelengths that are able to be reflected off of those dyes and pigments. So here we see a display of clothes in incandescent light, and we see that exact same display in a GE cool white light, and you can see it's become much less red and orange and much more bluish in appearance, right? So companies like Milliken that make dyes and pigments are very concerned about this, interested in this, because they don't want the shirt that you buy in the store to appear a different color when you walk out of the store into daylight. And you're like, wow, this isn't as purple as I thought it was going to be. And so they have things called light boxes that they can actually put dyed fabric in and then change the source of the light to mimic fluorescent light or mimic natural sunlight or mimic incandescent light, so they can see how this all looks. I'll tell you a trick that grocery stores use. The next time that you're in a grocery store, and I want you to go to the produce section, and I want you to look at the lighting in the produce section. Almost all grocery stores have different lighting, halogen lighting, that give more of this reddish, orangish color in the produce section to make the fruit and the vegetables and to make them appear healthier, right, than they would appear in just white light. And so that nice red apple that you buy at the store, when you get it home and look at it under a fluorescent light, may appear much less red. So next time you're in a grocery store, look, even in like Super Walmart, just above the produce section, they have special lights put in. It's really funny. Okay, color theory. Let's talk about how all this happens. We've already been talking about neural integration and the three different types of photopigments in the cones. And way back in the 1800s, just based on psychophysical data. So what does that mean? Psychophysical data means that we're going to change a stimulus and then ask a human, okay, what is your perception? And by doing these types of experiments, people were able to come up with the trichromatic theory, which is this theory that we have three selectively sensitive photopigments that are blue, green, and red, and they came up with this because of the ability to color match using those primary wavelengths of light. The other one is opponent process. They also observed that, hey, if you mix blue and yellow together, light together, it cancels out, this hue cancellation. And so they came up with this idea that there are opponents of color, red versus green, blue versus yellow. And that also turned out to be true. So both of these theories turn out to be true. We have neural evidence to support both of these. Trichromatic at the level of the retina, the signals coming out of the cones, and opponent process. By the time we get to the LGN, the thalamus, we now have blue versus yellow and red versus green. And we'll talk about how all that happens. All right, so just before we start, let's just go over our wavelengths of light so we can sort of agree that we're going to call blue 450, green 510, yellow 580, and red 600. All right, so as I mentioned, people were able to deduce the trichromatic receptor theory that we have three receptors, three different photopigments in the cones, by experiments with the blue, green, and red lights, showing that we can mix combinations of those to mimic any single monochromatic wavelength of light. And so that's because the blue wavelengths of light are activating short cones, the green are activating medium, and the red are activating long. And so we're essentially putting neural inputs into those three different types of receptors. So if you go to the textbook website, this has a nice little demonstration that I have a screenshot of here, where you can pick any wavelength of light and it shows you the activation pattern of the short, medium, and long wavelengths of cones. Here's an example. So the example we have down here in the corner is blue. And so in blue, we have a strong activation of the short, as represented by the bigger cone triangle, big activation of blue, very little of medium and long wavelength. And so that equals blue. Green, we're going to have primarily medium activation. Red, we're going to have primarily long wavelength activation. So we can have other colors like we've already talked about. Yellow would be equal activation of medium and long wavelength because it's right here, right? And it would produce equal activation of those cones. White would be equal activation of everything. And here's our favorite color, purple, which is going to be equal activation of short and long wavelength cones. So don't you see now how a single wavelength of light couldn't do that? Couldn't activate both short and long, just one wavelength? You have to have two in order to generate the color purple. All right, so here is a test field of yellow on the left. And now I'm going to take equal amounts of red and green, and when I overlap them, we generate the same color. We can't perceive the difference. Right here, this single wavelength of light is not being made here. We're seeing the combination of that. And that's because on the left, with 580, we get a pattern like this out of the cones. So we're going to say very little short, only one action potential, right? Pretty equal. It's maybe a little bit towards the oranger side. So we have 8 coming out of long and 5 coming out of medium. Now, if I take two wavelengths of light, green, which green would produce this 5 signal, but not the 8 or the 1. Red, which is going to produce the 8, we now have the same exact pattern with 530 and 620 wavelengths of light. We have the same pattern of output from the cones. So do you see how I'm saying that we're getting neural integration, right? So the two wavelengths of light are being added together to create a pattern of output from the short, medium, and long wavelength. And we can't discriminate the difference. This is exactly the same neural output as a single wavelength of yellow light. All right. What did we say about the vision system? Nothing's ever simple. It's always complicated, right? So do you remember when we talked about on, off, on center, off center, center surround receptive fields in the retina? Well, when we start to add in color, it gets a lot more complex. So it turns out that in the M cells, which come from the rods, which only have one photopigment, we do have this sort of like black and white on. Light excites the center, light inhibits the surround, or vice versa, right? But when we're talking about the cones, we're now talking about specific short, medium, or long wavelength cones. And so we begin to have opponent processes that are where if the center is on and red, then the surround is off, but green. So green is going to inhibit the surround. And if green is on in the center, then the opponent, red, will be off in the surround, right? And the same thing with blue and yellow, right? That's all I'm going to talk about that right now because it starts getting really complicated. We'll come back to that when we talk about color coding in the cortex. When we talk about the blobs and V4 a little bit. Because they're going to get the signals, the outputs from these center surround opponent processes. But just know that this is going on in that convergence of the signal to get from all those 120 million receptors down to 1 million inputs into the brain. Okay, so some perceptual evidence of trichromatic theory. If I shine this square of blue light, I want you to stare right in the middle of the blue light. Okay, so think about what's going on as you stare at this blue square, right? Staring right in the middle of the blue square. We are stimulating just your short wavelength cones. We're not stimulating the medium and long wavelength because it's pure blue light. So when we're stimulating those short cones, keep staring at the center. When we're stimulating those blue cones, we're bleaching them. We're using up the photopigment, right? So now if I show you white light, you should see a after image of yellow where the blue square was. Because think about it, we should be equally stimulating short, medium, and long wavelengths of cones, but the blue became fatigued, right? So having that square on there made the photopigment drop in the short wavelength cones compared to the medium and long. And so when I stimulate all of them, we're getting a bigger signal from medium and long wavelength. And so you see that yellowish afterglow. And as the yellow afterglow faded, right, that was the medium and long wavelength cones being stimulated enough that they come down to the same level as the blue. And once they're all equal, it becomes white. It's no longer yellow in tint. Here's another image. So stare right in the middle of the weird flag. And so this flag has been colored with the complementary colors of red, white, and blue, so that when I flash the white, you should see an after image of the regular red, white, and blue flag. Okay, opponent process evidence. We already talked about hue cancellation, right? So right here in the middle, where we have equal amounts of yellow and blue, they cancel each other out. So that's that opponent process, right? All right. Looking at the biology, right? We have three different photopigments, short, medium, and long wavelength photopigments, right? Those three outputs are going to form excitatory and inhibitory connections by the time they get to the LGN. So here we have a LGN neuron, and we see that when we have green wavelengths of light, it's inhibited. When we have red wavelengths of light, it's excited. And when we have an intermediate in between red and green, it's canceling out. It's the same as its baseline, right? And so we lose that color detection. So this would be an example of the center of this opponent process where we have red is excitatory, green is inhibitory. So that results in these, it results in four different signals now. So we go from three photopigments, blue, green, and red, now forming excitatory and inhibitory connections. We get a blue on, so excitatory from the short, and yellow inhibited. So we have two inputs from the medium and the long wavelength cones. That produces yellow inhibited. We have a green versus red. We have a yellow where we're getting excitatory input, equal excitatory input from medium and long wavelength, inhibitory blue, and then we have a red inhibited by green. All right. And this just shows you those connections a little bit bigger so that you can see the excitatory and inhibitory. Now, if the center of one, this is called double opponent process, right? If the center is excited by red, then it's always going to be inhibited by green. And the surround then will be the opposite, excited by green, inhibited by red. So if we know this, you should be able to predict what the neural activity would look like for each of these four different types of LGN neurons. So for instance, for a blue on, yellow off, we'll have more action potentials for blue, same as baseline for green and red, and inhibited by yellow. So green on, red off, yellow on, blue off, red on, green off. Right? It turns out that the inhibitory signals actually are coming from amacrine cells. So you remember amacrine cells? So what were amacrine cells involved with? Right, the convergence of the signal, making those center surround receptive fields. So the amacrine cells are responsible for the inhibitory inputs in here, right? And so I like this model because it shows you how yellow inhibitory can be created by having medium and long wavelength cones exciting that amacrine cell that's going to provide the inhibition, and then short is providing the excitation, right? What's sort of incomplete about this model is over here, we really need another amacrine cell pasted in on this side. The medium doesn't inhibit that. There's an amacrine cell. The medium is exciting an inhibitory amacrine cell to produce that green inhibition. All right. Why do we have opponent processing? Well, it allows us better discrimination. For one thing, we go from having 3 signals, blue, green, and red, to having 4 signals, blue, green, yellow, and red, right? And the opponent processing part actually creates more separation. The fact that green is in between yellow and blue, the opponents, and that yellow is in between red and green creates more separation. So if we look at the cone stimulation for green light, we get more stimulation from the medium and less from the long. And if we look at cone stimulation for red, we get more from the long and less from the green. And so if we were to graph those levels for 1 and 2, it would look like this, right? So our brain would have to process the difference between the medium and the long output to know that this pattern is green and that this pattern is red. But if we add in, that would be just with trichromatic, right? But if we add in opponent processing, now for a red on, green off center, we now, with green, have an inhibitory signal, which is very different than the excitatory signal for red. Much bigger difference here than trying to detect the subtle difference here. And that's going to allow us better discrimination. So which theory is right? Well, both of them, right? The level of the retina, we have trichromatic, color matching. The level of the opponent process cells, we get after images and simultaneous contrast as psychophysical evidence that blue versus yellow and red versus green. All right, color coding in the brain. Here's where it gets even more complicated, if you can believe that. The return of the blobs. So you remember when we talked about V1, we said, okay, we have these hypercolumns, there's angles of lines from horizontal all the way to vertical, stretch across the hypercolumn. And then on top of that, we're going to throw blobs of color. And we called them blobs because it wasn't like the whole hypercolumn was red and the whole hypercolumn was green next to it. They were just sort of splotchy blobs. And it's really complex. We know that that's the way it is because we can map it. We don't exactly know why it's that way, why it isn't sort of orderly, but our brain is able to make sense out of this and take those red signals and send them to V4 so that we can see the colors that different shapes have. So V4, specialized area of secondary cortex responding to colors instead of shapes or movement. And so somehow we're able to take these overlapping receptive fields, just like we talked about before, to generate the horizontal, all the different angles of lines, right? This case, this would be a red horizontal line surrounded by a red inhibitory surround, right? Or a green excitatory surround with a green inhibitory in the middle. So for that example, these are the different types of signals that we could get. And this is called a double opponent process because we're coding for both red and green in the center. It's just red is excitatory and green is inhibitory. And we're coding for red and green in the surround, where red is inhibitory and now green is excitatory in the surround. Pretty crazy. Told you it gets complicated, right? All I really want you to understand is that is the organization at V1 is in these blobs. It gets better organized into these center surround receptive fields, double opponent process, red versus green, blue versus yellow in V4. Okay, let's take a minute and talk about color deficiencies, right? Color blindness. Color blindness occurs when we have a deficit in one of those cone photopigments. And there's two main sort of cases that we call dichromatism, where you have two photopigments instead of three. And the most common of those are deuteranopes and protonopes. So protonopes lack the L photopigment. Deuteranopes lack the M photopigment, the medium photopigment. It's very rare to get trentinopia, which is where you lack the short cone input. So here's what I want to say about that. If somebody is a protonope, let's focus on that, right? There may be some, like, because of the overlap of medium and long wavelength cones, there may be some reds that they can't detect. But orange is stimulating the medium, so is yellow. And so they can detect that light. They just detect it in a different way than somebody with three photopigments would detect it. So they can detect short and they can detect medium, and they see sort of this change from yellows into hue cancellation into blues, right? And so they learn, they grow up in this condition. So when they're learning their colors, they recognize this very bright yellow as orange. That's orange to them. That's what orange looks like to them. And they're able to discriminate. It's harder for them maybe to learn that yellow is a slightly dimmer hue and that green is this very pale yellow and that blue is blue, right? And if they had a very long wavelength color that was purely activating the long cones that they don't have, then what color would that look like to them? Someone would say, this is red. What color would it look like? It would be black to them, right? It would lack color because there's no stimulation of the cones, right? So it's the absence of stimulation. Isn't that wild? And they would say, oh, that's red, right? And they would have a difficult time discriminating between red and black. All right, so let's watch this video that sort of talks about our unique color perceptions.
Okay, so we both know that this apple is red, but is what I see as red the same color that you see as blue? This is going to get complicated. Hold on a second. Anthony here for D News, and Mr. Nick VDB asked, could you do an episode on how we can tell that the colors I see are the same ones that you see? Now, this is something that I think everyone wonders at some point. We both know that the patches on the wall behind me are blue and orange, but does blue to you look the same way that red does to me? Now, obviously there are issues here, physical issues like color blindness that would keep someone from perceiving colors exactly like other people, but let's put those aside and say that we're dealing with a standard human eye. Now, we see colors because of rods and cones in our eyes. And we've got three types of cone cells for seeing red, green, and blue. A lot of other mammals only have two kinds for seeing blue and yellow. Birds have four and can see all three of those colors and ultraviolet light, which is one of the reasons that birds are so creepy. Anyway, so you'd assume that since all the hardware of the human eye is the same, and it's all based on specific wavelengths and colors of light, that we would all see things the same way within a small margin of error. But here's the thing. The number of cones in people's eyes vary hugely, and the kinds of cones aren't evenly distributed. So one person may have mostly red cones, while someone else has more blue, which means that the brain is more instrumental in actually decoding color. It steps in, analyzes the information, and helps us sort everything out. And that introduces a lot of doubt. See, our brains are these pattern-making machines that pick up a lot of slack for us. They're constantly filling in missing information. They're making educated guesses to keep us running smoothly throughout the day. And here's something interesting about color. The more you learn about it, the more you can see variations in it. For instance, check out this ring of green squares shown to people by the American Psychological Association. One of those colors is different, ever so slightly. Can you tell? You might be able to, according to the study, if you're an artist or a designer or you have some other career that requires a honed sense of color. It's this one, by the way. But wait, what about studies on the effects of color on our emotions? Trace talked about them in a video, how colors affect our mood and how food tastes, things like that. Orange makes us energized, blue makes us calm. So how does that happen the same to everyone if we see colors differently? Well, welcome to the biological part of seeing color. We're diurnal creatures, right? We're awake during the day, we're asleep at night, and that's because of light. It's bright out, we wake up, yes? Well, what if I told you we aren't reacting to brightness as much as we're reacting to the color of light? A study in the Journal of Animal Behavior found that diurnal animals sleep better in bright blue light than in dim yellow light. When do we see blue light? Late day, evening. Yellow light comes in the morning and midday. Now think about what I said earlier, how those other mammals only have those yellow and blue cone cells in their eyes. Well, isn't that convenient? Just the right equipment to tell when to be tired. Now that means we're all biologically wired to respond to the color spectrum in the same way. It doesn't matter if you and I perceive yellow the same. Our bodies know that seeing it means it's time to be energized. Okay, so bottom line, do we see colors the same way or not? For a really long time, science has sort of taken this behaviorism stance to it, which basically means, hey look, if we're all responding the same way to the same thing, it's not an issue if we all perceive it slightly differently. Let's all just let it go. But chances are there is some variation. Based on everything I've read, my hunch is that the differences in color perception aren't too crazy. What do you guys think? Is my red your green? Vice versa?
All right, pretty cool stuff. So dichromatism happens when there's a mutation on the gene that's supposed to produce that photopigment, right? Those genes are found on the X chromosome. So this makes it a sex-linked disorder because remember, females have two X chromosomes, so they would have to have a mutation on both X chromosomes. If they only had a mutation on one, then the non-mutated version of the gene on the other X chromosome could produce the photopigment. So deuteranopes and protanopes are the most common types, as I mentioned. There's about an 8% prevalence in males because males only have one X chromosome. So if that mutation happens on that gene, they don't have a backup. They won't make that photopigment. Because females have the backup, it goes from 8% to less than 0.5% prevalence in females. And so as I mentioned, deuteranopes or protanopes would still see shades, variations. They could still discriminate between colors, but they would have a hard time discriminating between red and green because they're very close together in that spectrum of yellow that we saw. So they wouldn't be able to see the 74 here in the middle. So this would be a type of test. We're going to do color vision testing when we get back in person. That's going to be your next lab. And I've actually had some people discover that they were colorblind by doing this lab, some male students who didn't realize that they were colorblind because they can still discriminate between colors. They just see them in a different way in their brain. All right, we can also have cortical deficits in color vision. If you could have monochromatism, which would be literal color blindness, only being able to see in shades of gray, right? That's a very rare disorder. You would have to have only one cone photopigment, right? Cortical color blindness is pretty rare because of the way it's organized, right? So blobs in V1 are going to not only be color blindness, but blindness, right? You're not going to be able to see those shapes if you have damage in V1. It'd be very rare to just have damage in V4, to have a lesion or a tumor or a stroke that only affects V4 and doesn't affect other things. It can happen, but usually there's other areas of the brain that are also affected. So it's usually combined with other deficits. So, damage can be caused by lots of different things. I mentioned strokes, but multiple sclerosis, where you could have demyelination in that area that would cause problems with color vision. You could have things like that affect the oxygen or glucose supply, like anemia or liver disease. Okay, getting towards the end here. Two more topics I want to briefly touch on. I want you to look in your textbook on the section on color constancy, and I want you to work through this on your own. Okay, I want you to do some critical thinking to understand this. But essentially what we're talking about is if we change the illumination, the type of light. So here we have white light and we have a green and a gray patch. If I now change the color in the room to red light, you're still going to associate this with green and white, but the input to your cones is going to be very different. In fact, if we were to just isolate and cover that up, then this would look very red, but if we put it in context of the illumination of the red light, then it appears white. We associate that as white. Okay? So go through that section of the book and work through that and make sure that you understand color constancy. The other section I want you to work on in the textbook is the color vision in animals, right? So we just saw in that last video that birds can actually see have 4 cones and can see ultraviolet light. There are other animals that can see the other side of the spectrum, can see infrared light. Right? So we're going to watch this last video, and then I'll have an Ed puzzle question for you. We'll watch this last video on color vision in animals.
Right now, there are animals out there seeing colors that you cannot even conceptualize. Biology is crazy unfair sometimes. Anthony here for D News, and researchers just found out that rats constantly keep an eye towards the sky to avoid predators. Each eye actually rotates independently and sees something different, specifically so they can keep watch for danger. Horizontally, their eyes can be at about 40 degrees of difference. Vertically, they can go about 60 degrees cross-eyed. Now you compare that to us. If we even go 1 degree out of our usual alignment, we get double vision. Guys, can we get real? The human eye is amazing, but things get so much more crazy in the animal kingdom. We mentioned in another video how we see colors. We've got these rods and cones, right? Rods take in brightness and motion. Cones are what perceive color, and we've got 3 of them, which allow us to see red, blue, and green light. We also have binocular vision, right? 2 eyes on the front of our heads with overlapping fields of view. Pretty slick system. Compare that to cats and dogs, who only have 2 cones. They see combinations of blue and green. But by giving up those cones, they have more rods, which allow them to sense motion better. Cats also have a special layer of cells in the back of their eyes called the tapetum lucidum, which gives them night vision that is 6 times stronger than ours, which is like lame-o stupid vision to a leaf-tailed gecko, because they can see 350 times better than us at night thanks to their vertical pupils and the fact that they only have cones in their eyes. 3 types of supersized cones that let them see color clearly in the dark. The eyes of nocturnal geckos also have these concentric zones that refract light differently. They have multi-focus eyes. Scientists are actually studying them to create new kinds of cameras and even multifocal contact lenses. But inarguably, the best, most amazing eyes in the world belong to that wonder of wonders, the mantis shrimp. I love you, mantis shrimp. So we humans have got our 3 cones, red, green, blue. Mantis shrimp have compound eyes with 16 color receptors. They see ultraviolet, infrared, polarized light. They're like a tiny covert military operative. They can even see circular polarized light, which is a type of light no other living thing on Earth can see. What does it look like? I don't know. I cannot describe it to you. Nothing can see it. You know what sees circular polarized light? DVD players. Only the mantis shrimp sees them better. Their eyes are also on stalks that move independently, so they're taking in both views at once, like mice and lizards, but they also have trinocular vision. You could destroy a mantis shrimp's eye, and it would totally still have full depth perception in the other one. I mean, that is, if you could get to it before it attacked you with its tiny claws that move at the speed of a bullet, boiling the water around them. I mean, we've seen the oatmeal comic, right? God, I love the mantis shrimp. But anyway, yes, animal vision. There are so many crazy examples I did not even touch on. So tell me about your favorite animal vision powers down below, and subscribe for more D News.
All right. Man, that guy was really jacked up on some mantis shrimp. He may or may not have a problem. Here's your review points on color vision. We have 2 pages of them. Please be learning this information as you go. Okay, this is the first lecture in exam 2 section. But we only have 4 lectures for exam 2. So we're going to be having a exam 1 when we get back, the week we get back, and then the next week we'll have just 1 lecture and then exam 2. So they're going to come, compacted, but we're going to do the best that we can. We're going to make this work. And so make sure that you are staying on top of the material. All right, I'm going to try and get your other lectures recorded and posted by the time class happens. I had actually, the second time I've done this lecture. The first time I did it, my microphone wasn't on, so that was fun. Got to do a practice lecture for you. But I'll get better at this, and make sure you do this Ed puzzle question.